Hello, dear students. My name is Francesco Tamagnini, and today we will talk about the final part of the renal system function, which is the ability of this system, this amazing system, to regulate the pH of our body fluid. So at the end of today's lecture, hopefully you will be able to understand the chemical processes underlying the acid-base balance, so the buffering of the plasma pH, to understand what buffer means, what buffering capacity is, and how this can be calculated using the Henderson-Hasselbalch equation. You will be able to understand that the acid-base balance is indeed an integrated process which involves different and several organ systems. Understanding and in particular understand how the renal system can regulate the pH of the plasma, understand how the acid-base disorders can arise, and the role of both kidney and the respiratory system, how they can contribute to or compensate for these disorders, and how measuring the plasma pH and bicarbonate levels can be used as a diagnostic tool to diagnose renal disturbances and disorders. So I will start with an introduction and a discussion of the pH buffering and buffering systems in the blood plasma. I will then switch to the integrated acid-base balance, focusing on the renal regulation of the acid-base balance. And then finally, I will talk about acid-base disorders, taking some case studies as example and how they are diagnosed. And finally, I will end up with an overall summary. So the renal system is involved also in the homeostasis of pH in body fluids. Now, the renal system doesn't do this alone, is obviously tightly integrated with the cardiovascular and the respiratory system. So the acid-base homeostasis is interdependent and integrative, which involves different body systems. And so it's important to reinforce the concept that the homeostasis is maintained by the whole body and not just by specialized organs. By the whole body, thanks to the activity of specialized organs and systems working together. So the pH is the co-logarithm of the concentration of protons in a solution. This is a definition that I've learned in junior high and for some reason just got stuck into my head. So the co-logarithm is the minus of the logarithm of a concentration. Okay, so the value of the pH can vary between 0 and 14, 15. There's really no limit to the numbers, right? We do have superacids that go below 0 or below -1, which are called superacids, or we can have superbases that go above 14. But most of the times, in our life, we're going to experience pHs, whether in cleaning products or in chemical reagents, if you work in a lab, that go between -1 and 14. Pure water, which has an absolute acid-base balance, has a pH of 7. Means that the pH is neutral, which means that the concentration of hydrogen ions is counterbalanced by the concentration of hydroxylic ions. This is, of course, a very old concept of acidity. We can also have different, definitions of acids and bases, right? The Bronsted-Lowry, the Lewis um, definition of acid is of being an acceptor of electron doublets, while the base is a donor of electron doublets. But in more general terms, the oldest definition of acid is what generates, what releases protons in a solution, and the oldest definition of a base is what leads to the release of hydroxylic groups, hydroxylic anions in solution. So the co-logarithm of the concentration of protons is a measure of the acidity of a solution. So Søren Sørensen, who's a chemist who originally started to work for Carlsberg, invented the log scale for measurement of the alkalinity or acidity of a solution. Probably is the best log scale in the world, or definitely the best known logarithmic scale in the world. Okay, so after these anecdotes, if you want to look at Søren Sørensen, you can, I don't know, Google it and look a bit at the history of the pH and of Carlsberg beer and how, this very important tool for every chemist and physiologist and doctor and pharmacist in the world that is coming from the processing of beer. Anyways, the process that regulates the hydrogen ion concentration or protons in the body fluids within normal limits and measured in terms of pH is known as acid-base balance. Okay, so pH is a negative logarithm, as I said, or co-logarithm. I should have specified in base 10 of the hydrogen ion concentration. Okay, so pH is equal to the co-logarithm of the concentration of hydrogen ions. So the concentration of protons in the blood plasma is around 4 times 10 to the power of -5 milliequivalents per liter, which is around 7.4, which is not exactly neutral, but it's very close to neutrality. And this parameter is highly restricted. Okay, so even small changes in few decimal points of pH can have, pathogenic consequences and even lethal consequences for the organism. Okay, so if the concentration, for example, if the pH of the plasma falls 7.22, we are in the situation which is known as acidosis. If it goes to 7.7, we are in the situation that is known as alkalosis. So the range is between 7.3 and 7.5. Below and above, we have, pathogenic ranges. Below 6.9 and above 8, we have death. So almost all fluids in the body have a strictly regulated in a pH of 7.4. Okay, so this is true for the internal environment, so extracellular fluid, interstitial fluid, plasma, and also for the intracellular fluid. Okay, so there may be slight variations, but most of the interstitial fluids and intracellular fluids and plasma fluids have a pH of 7.4. Urine, on the other hand, has a variable pH with an average around 4.5. The truth is that the urine's pH is often or almost always negative because we're animals. So we do have a catabolic metabolism, or an oxidative metabolism, which leads to the production of energy by the oxidation of glucose. And the oxidation of glucose leads to the production of carbonic acid or CO2 in gas and water. So it's an acidogenic metabolism, as in opposite to the metabolism of plants, which is an alkali-producing metabolism. So our metabolism produces protons, okay? To survive, we need to produce protons, which means that fairly quickly, the tissues in our body would become acidic if we don't have means for regulating the acid-base balance. And the long-term maintenance of the acid-base balance is maintained by the kidneys, which generate urine, which contains an excess of protons, which are removed from our body, okay? So the acid-base regulation is an integrative process which, as the regulation of, the blood pressure occurs at many levels, at a local level and in the short term, at a more generalized level in the mid-term, and at a fully generalized level in the long term, okay? So the buffering of pH is also known as the maintenance of pH within certain desired levels. Okay? So the first form of buffering of pH, so the first form of buffering of protons, occurs at a very local level in the inside of the cells, in the intracellular fluid, in the cytoplasm of a cell, or in the interstitial fluid. Inside of the cells occurs by the means of proteins, phosphates, and bicarbonate, which bind protons and make them bio-unavailable. And this occurs inside of the cells, but it also occurs outside of the cells in the interstitial fluid. And then a mid-term regulation of acid-base balance by the excretion of CO2 via the respiratory system. So increasing the respiratory rate leads to the increased removal of CO2, which means increased removal of carbonic acid from the blood, which means a decrease in the pH of the blood. And then finally, in the longer term, the excretion of protons via the renal system, via urines, which may take. Okay? So we have a buffering system that is immediate and localized at the level of the buffering systems provided by proteins, bicarbonate and phosphate, and then a midterm system, which acts within minutes. So the excretion of CO2 via the respiratory system, and finally the long-term maintenance of the acid-base balance via the renal excretion of protons. So one example of protein buffering system is the hemoglobin. Hemoglobin can bind protons. Also, proteins contain both acidic and basic sites. So they contain, of course, they're aminoacidic, right? So they've got an amino term and a carboxy term, and the carboxy term is basic, while the amino term is acidic within a protein. And this can bind or release protons. Also phosphates, which are freely available in the extracellular and intracellular space, but mostly in the extracellular space, are involved in the buffering of pH because they can bind protons. Because the phosphate bound to a proton represent an weak acid-weak base conjugate pair. So this is important because phosphates represent a urinary buffer and intracellular buffer. So also bicarbonate represents a very important buffering system. The CO2 produced by the catabolism of the organism dissolved in water generates carbonic acid, carbonic acid is dissociated into protons and bicarbonate. Bicarbonate can bind protons, making the protons unavailable and reducing the pH or buffering the pH. The bicarbonate is the main proton buffering system in the extracellular fluid and in the intracellular fluid. And of course, high concentrations of CO2 will move the balance to the right, leading to increased production of protons and increased release of production, reducing the blood pH. High concentrations of bicarbonate will drive the equilibrium to the left, leading to the increased levels of undissociated carbonic acid and, of course, CO2. So the excretion of CO2 via increased rate of expiration will remove protons from the blood and increase the level of the pH in the blood. So the buffering capacity, of course, depends on many factors. The first of all is the concentration of buffer. Higher the concentration, the greater is the buffering capacity, but also by the pKa, which is the apparent dissociation constant of the buffer. So the closer the buffer pKa is to the plasma pH, the best is the efficacy of the buffer for homeostasis. So in order to calculate that, we have the Henderson-Hasselbalch equation, which is used to estimate the pH of a buffer into the solution, to find the equilibrium pH in the reactions which involve acid and base. So if it's towards the base or towards the acid, and also the isoelectric point of proteins, which is the pH at which the negative charges on a protein are the same as the positive charges in a protein. So it's the pH at which the net charge on a protein is equal to 0. So Henderson and Hasselbalch are the two scientists responsible for generating an equation that will allow us to predict the pH of a solution depending on the concentration and the dissociation properties of a compound. So we have a compound which contains protons, okay? If the proton is not dissociated from the rest of the molecule, it will not be released in solution. So the pH of the substance will not decrease. But if the proton is dissociated from the anion, then the pH will decrease. So this is obviously a balance, right? It's an equilibrium. And how do I predict the pH? So the concentration of protons into a solution as a function of the concentration of the compound and the dissociation constant of the compound. Because of this equation, which states that the pH is equal to a constant, which is the apparent dissociation constant of the acid, plus the logarithm of the concentration of the dissociated anion divided by the concentration of the undissociated substance.